All right. Hena, we will start from this question. I think 2019 වල MCQs උත් අපිට තියෙනවා ටිකක් discuss කරන්න. I am planning to have two extra classes next week. One I was thinking tomorrow, Monday 7 to 10. The other one is Wednesday 7 to 10. What do you feel about it? Is that okay? Tomorrow 7 to 10 and Wednesday 7 to 10. මේ දවස්වල y'all anyways home නේද? No, you have school. But are the term tests over? එහෙනම් there is a bit of an issue. Saturday class එකෙත් මට ඒක කීප දෙනෙක් කිව්වා test එක ඉවරයි. But if you have school, I can't do a class. That's not right. I will check and confirm with you. You have term tests. Some of you have term tests. Even in the Saturday class, Onilka told me that they have term tests in the coming week. මම හමුණු චුට්ටක් බාලා කියන්න ළමයි. කොහොමද ඊයේ උදේ class එක කරන්න බැරි වුණා. So they will have a rescheduled class for the class they missed yesterday. But extra classes wise, I will let you know what to do. Okay. An outline of the classification of living organisms is given below. Living organisms are classified as archaea, bacteria, and what is W? What is W? Others? Now I need all of your answers. As usual, you know how past paper revision we do, right? Not mammalia. What are the main three domains? अब मैं discuss करने question එකක් නෙවෙයි නේ. The main three domains are archaea, bacteria, and eukarya. So it is eukarya. If you take lamai, all organisms, all living organisms on this earth, මුලින්ම divide වෙන්නේ domains තුනකට. There's domain archaea, domain bacteria, and domain eukarya. Domain bacteria වල ඉන්නේ all the bacteria, they can live anywhere and everywhere. And you know that bacteria ගත්තොත් they are sensitive to antibiotics, right? Archaea වල තියෙන්නේ these organisms live in extreme environmental conditions in archaea, right? Like volcanoes, hot springs, deserts, ice caps and glaciers, right? And ඒ වගේ හරිම extreme normal organisms වලට survive වෙන්න බැරි extreme conditions වල තමයි archaea ඉන්නේ. Eukarya කියන්නේ ඒ දෙක අරගෙන අනිත් කට්ටිය ඔක්කොම. Then if you take the domain eukarya, these are domains, domain archaea, domain bacteria, domain eukarya. Bacteria domain එක that is sensitive to antibiotics. ඒකයිනේ අපිට when we get a bacterial infection, the prescribes an antibiotic course. Antibiotic course එකක් දෙනවා නේද bacterial infection එකක් තියෙද්දි. That is because antibiotics can kill bacteria. But antibiotics cannot kill archaea or eukarya. So archaea and eukarya are not sensitive to antibiotics. Bacteria is sensitive to antibiotics. So eukarya wala hatarak innawa. There is protista, there is fungi, there is plantae, and finally what is it? Protista, fungi, plantae, and animalia. Animalia wala. Now protista wala inne kawuda? Some examples tell me. Protista has two main types of organisms. What are they? Protista has two main types of organisms. What are they? Protista wala bacteria na. Bacteria inne domain bacteria wala. Now protista is under eukarya. What are the organisms in protista? Yes. Okay. Paramecium is one example. ළමයා අපි revision වල කොච්චර කතා කරලා තියෙනවද protista ගැන? I mean, in under classification lesson, we have discussed a lot in the revision overall. Yes. If you take kingdom protista, there are two types of organisms. මේක අපි already ඉගෙනගෙන තියෙන්නේ. මොනවාද two types? One is we have algae. Algae කියන්නේ ඒගොල්ලන් තනියෙන් photosynthesis කරනවා, and also they are like in aquatic environments, they are the primary producers. ඒ කියන්නේ ponds වල lakes වල එහෙම ඒගොල්ලන් තමයි ගස් වගේ ඉන්නේ. Now අපේ නම් there are multiple trees now in the terrestrial environment. In the aquatic environment there are trees of course, but there's also algae. Okay, so they are primary producers in aquatic food chains. And then you have seen like an example would be ulva. Very good. Ulva is an, since I'm typing it, I wrote it in italicized font. Samane underline karna. Underline kare neta tawulak nahi megolange na mukada these are just one word letters. But hari nang underline karna one. Then what are the other types of organisms in protista other than algae? Algae arena tawa set එකක් ඉන්නවා. Algae arena tawa set එකක් ඉන්නවා. What are they? Examples are කමක් නැහැ. Protozoa. Protozoa කියන්නේ they are not photosynthetic, they are heterotrophs. Examples අපි ඉගෙන ගත්ත paramecium, yes. Amoeba, sorry spellings are wrong. Amoeba. I මොනවාද? Paramecium. Paramecium කියන්නේ මෙන්න මෙහෙම එක්කෙනෙක්. මෙහෙම චුටි. If I try to draw it, it will take forever. මට මේක අඳින්න නැත්te අමාරුනේ cilia වගේ මෙහෙම චුටි. These are called cilia. Small fibers are, small hairs are all around the body of paramecium. Then amoeba is just like a random body with ඔන්නෝම නිකන් තියෙන random එකනේ. තව if you remember there is euglena, Chlamydomonas. These are some examples for protozoa. So kingdom protista වල ඉන්නේ there's algae and there's protozoa. They are kind of different from each other. Okay, එතන fungi වල ඉන්නේ කවුද? Fungi. Right. So when we talk about fungi, we know fungi can be unicellular or multicellular. Examples අපි ගත්තොත් එහෙම yeast is a fungi. ඊට පස්සේ penicillium is a fungi. ඊට පස්සේ අපි ගත්තොත් mushroom කියන්නේ fungi එකක part එකක්, right? And also when we talk about fungi, we know there are so many useful effects, like useful things from fungi. What are the useful things of fungi, Lamai? Tell me different ways how fungi can be useful. Yes? To produce antibiotics. Antibiotics are what is used to destroy bacteria. To make some of these antibiotics, we use fungi. Yes. To make bread, alcohol. මොකද bread alcohol වගේව ferment කරනවානේ. දැන් alcohol උනත් let's say a wine එකක් හදාදී ඒ මොකක් හරි base කරන fruit එක. Let's say grapes. ඒ grapes වල spirit එක අරගෙන then they let it ferment. And ඒතර ඒකලා ferment කරන්න කලින් ඒකට fungi එකක් use කරනවා. Okay, and then they let it ferment for years and years. ඔයගලන් දකින wine cellars ඔය පරණ movies වල එහෙම තියන්නේ. Wine cellars. So ඒ වගේ wine ගොඩක් කල් ferment කරනවා. And if you take like ගොඩක් summer alcohols ගොඩක් ඒවා එක එක grains වලින් හදන්නේ. Barley, maize, එහෙම grains වලින් හදලා they let it ferment. So ඒතර ferment කරද්දී ඒගොල්ලන් yeast දානවා. I got some other answers in the chat box. Decomposed dead bodies. Good. Why? Decomposed dead bodies can lamai, let's say a bacteria or a fungus. බැක්ටීරියා හරි ෆංගයි එකක් හරි තමයි මොකක් හරි dead plant එකක් හරි animal එකක් හරි තියෙනවා නම් ඒ body එක decompose කරන්න. For an example, let's say a plant falls down and dies. A dead plant. ඒක decompose වෙන්නේ නැත්නම් එතන dead plant එක forever තියෙයි. And there will be other plants dying and it will pile up. Just like that, humans may be, animals may be, when they die, if they don't decompose, හැමතැනම bodies තියෙයි. මම ගන්න කිව්වේ මේක එකපාරක්. ඒකින්ද it is very important to decompose. Decompose part එක කරන්නේ bacteria and fungi. So now fungi will useful එකක් තමයි decomposition. And also like food items හදනවා එරමෙව, right? What are the harmful effects of fungi? Causing diseases, okay. And yes, food spoilage. Bacteria and fungi එකතු වෙලා කරන හොඳ දෙයක් තමයි decomposition. Bacteria and fungi එකතු කරලා කරන නරක දෙයක් තමයි food spoilage. If you leave some food out in the open, after some time, it could be a few hours, it could be some days, it could be ඒ වගේ වෙලාවක්, and you come back, the food will be spoiled. So you cannot consume it further. That is because bacteria and fungi have worked. Plant ගැන නම් මම ආයි classification එක ගැන කතා කරන්නේ නැහැ වැඩි මොකද ඒ ගැන ගොඩක් සැරයක් කතා කරලා තියෙන්නේ නේ. අපි කිව්වා plant එක ගත්තොත් they are classified first of all as non-flowering and flowering. Non-flowering වල තියෙනවා seedless and seed plants. Seed plants වලට අපි කිව්වා gymnosperms කියලා, right? The seedless plants are examples අපි කිව්වා marcantia, pogonia, salvinia, drinaria, හැමෙම. ඊට පස්සේ non-flowering seed plants වලට which are gymnosperms, our examples are cycas and pinus. ගොඩක් වෙලාවට examples තමයි වැඩියෙන් අහන්නේ. So better to remember some examples. මොකද MCQ වලම හොඳට questions අහන තැනක් තමයි එතන. ඊළඟට flowering plants වලට generally අපි කියනවා angiosperms. In flowering plants, again, flowering plants can be divided into two. What are they? Give me a second, lamai. Okay. So, yes, flowering plants can be divided into two as monocots and dicots. So, monocots features api darnawa, dicots features, examples edaka mataka tiyaga. Then we are coming to animalia, which can be divided into two as invertebrates and vertebrates. What comes to Y? Y වලට මොකක්ද දෙන්නේ? Excuse me. Yes. Y වලට එන්නේ mammalia. Why vertebrates ගත්තාම vertebrates කියන්නේ animals that have a vertebral column. එතකොට ඒගොල්ලන්ට නිකන් bones තිබ්බට වැඩක් නැහැ. Vertebral column එකක් තමයි තියෙන්න ඕනේ. So Pisces is fish. Then amphibia is frogs and toads. Yeah, Chlamydomonas is algae. Okay, yes. Chlamydomonas man protozoa වලට දාලා තියෙන Chlamydomonas is an example of algae. Yes, that's correct. Ulva, Chlamydomonas, they are both algae. Paramecium, amoeba, euglena, they're all protozoa. Okay. Chlamydia monozoa දෙන්නම ළමයි green algae. Even in algae, ඔයගොල්ලෝ දැන් එච්චර ඒක ඕනේ නැහැ, but in A levels you will be learning there are 3 types of algae called green algae. ඔයගොල්ලෝ දැක්කොත් එන්නේ එකේ videos වල ඉන්නවා මේ documentaries වගේ ඔයගොල්ලෝ බලනවා නම් some algae they are red in color. So there is a type called red algae too. And there's another type of algae called brown algae. Brown algae වලට මේ ඔයගොල්ලෝ අහලා තියෙනවනේ kelps කියලා තියෙනවා. There are like huge brown algae forests in certain oceans. Can you ගොඩාක් උස්ස නිකන් forests වගේ brown algae තියෙනවා summer oceans වල. It's the same with green algae as well. But brown algae හැමතැනම නැහැ. සමහර තැන්වල තියෙනවා we call them kelps. Green algae තමයි photosynthesis කරන්න ඇත්තටම red algae and brown algae I mean they don't have chlorophyll. So Ulva, Chlamydia monas, they are both green in color. They are both types of green algae, but they are not like huge algae. They are considerable size. They are podi algae. Dekak tamai Ulva and Chlamydia monas. Good observation. Okay. So vertebrates wala, as I said, Pisces is fish. Reptilia amphibians are amphibians, right? Then reptilia can be reptiles like crocodiles, monitors, then geckos, lizards, whatever. Aves are birds and mammalia can be mammals. What comes to Z? Yes. Invertebrates lage Coelenterata is the same as Cnidaria. Samhara velawata lamai they can give the term Coelenterata. මම past paper සමහර ළමයික කරාදී Coelenterata දැක්කම they don't like the word because we are used to Cnidaria. But එහෙම කියලා හරියන්නේ නැහැ. Coelenterata is also another word for Cnidaria. Annelida, mollusca, arthropoda, and echinodermata. Name the domain to which living organisms not sensitive to antibiotics belong. What did you write for that? Not sensitive to antibiotics. Okay. Others? Yes. Bacteria is the only domain that is sensitive to antibiotics. So if they're asking the domain that is not sensitive to antibiotics, it's either archaea or eukarya, as I said previously. To which kingdom do algae belong? Now we know it is kingdom Protista. A characteristic of the invertebrate groups we have been given. Opposite to each characteristic, write the group of animals having that characteristic. Bearing soft bodies. Which animals bear soft bodies? Mollusca. There is no other answer for this. අපි සාමාන්‍යයෙන් invertebrates වගේ soft bodied animals කියන්නේ mollusca ලට. Division of the body into equal segments. Equal segments, huh? නැත්නම් arthropoda ලගෙත් divide වෙලා තියෙනවානේ segments තුනකට. There's head, we call it tagma, right? There's head, there's thorax and there's abdomen. හැබැයි මෙතන equal segments. That is Annelida. Annelida. Then existing in two forms, polyps and medusa. Cnidaria or Coelenterata. මෙතන Coelenterata කියලා දීලා තියෙන හින්දා I will write as Coelenterata. මොකද shown in the above classification මෙතන Cnidaria කියලා ලිව්වට ලකුණු දීලා තිබ්බා. But I'm just trying to be safe, right? To get the maximum marks. මොකක් හරි හින්දා ලකුණු කැපුවොත් කියලා මම Coelenterata කියලාම ලිව්වා. එහෙම තින් unreasonable විදිහට ලකුණු කපන්නේ, so don't worry about it. Is this part clear lamai? Give me a thumbs up. Okay. Then B එක. It is a photosynthesis question. A setup arranged by a student to study a factor essential for photosynthetic processes shown in the diagram. This is quite easy. අපේ textbook එකේ තිබ්බ වගේම setup එකක් තමයි use කරලා තියෙන්නේ නේ. Colorless glass strips, black glass strip, then the rest of the leaf is exposed. This has been kept in dark for 48 hours and then was exposed to light for 5 hours. Afterwards, the glass strips covering the leaf were removed and the leaf was tested for starch. To test which factor essential for photosynthesis was this setup used? What do you think? What is the factor? Okay, yes. Yes, factor is light. Light කියලා ලිව්වත් හරි, light energy කියලා ලිව්වත් හරි, sunlight කියලා ලිව්වත් හරි. Bigger sentence එකක් ලිව්වත් කමක් නැහැ. මෙතන fact එක විතරක් අහන්න ඉඳන් I'm only writing the factor. You can write to test whether light energy is used for photosynthesis. එහෙම මුළු එකම ලිව්වට කමක් නැහැ. What is the reason for keeping the setup in the dark for 48 hours? To remove the starch, because lamai, even if during the 5 hours it was exposed to sunlight, black glass stripping cover වෙච්ච තැන obviously photosynthesis උනේ නෑනේ. හැබැයි යා කලින් photosynthesis කළා store කරපු starch තියෙන්න පුළුවන් නේ. That is why we are keeping it in the dark for 48 hours, because during that 48 hours it will not do photosynthesis and it will use whatever the starch it might have kept storing, right? So to remove starch. To remove starch, what ati? Nathan, to remove stored starch. Or you can even write to de-starch. Any of this is accepted. Write the color that can be seen in each of the following parts of the leaf during the test for starch. Part covered with black glass strips. What is the color you can see? මෙතන ළමයි no color change කියලා ලිව්වොත් ලකුණු දෙන්න බෑ. Color change එකක් තියනවද නැද්ද කියලා නෙවෙයි ඒගොල්ලන් අහන්නේ. ඒගොල්ලන් අහන්නේ the color that can be seen. ඔයා මේක කරද්දී ඔයාට ඇස් දෙකට පේන color එක. Color change එකක් තියනවද නැද්ද කියන එක නෙවෙයි අහන්නේ. What is the color you can see? Black glass strip එක දාපු තැන ළමයි. First of all, when we are doing the starch test, the leaf will be like, leaf is usually a green color. Okay, ළමයි. So how do we do the starch test? මන්ට ඒක දැන් ඕනම එකක. When we are here we are testing for light energy. And if you remember, there's another setup where we cover 2 leaves in polythene bags. එකකට water දාලා අනිත් එකට KOH දානවා. ඒකේ අපි carbon dioxide test කරනවා. Then mosaic leaf එකක් අරගෙන අපි chlorophyll වලට test කරනවා. ඒ test කරද්දී අන්තිමට හැම එකෙම කරන්නේ starch test එකක් නේද? So starch test එකක් කරද්දී you know, our leaves are green. The leaf is green in color. Ne? The leaf is green in color. එතකොට මේකට මම iodine දාලානේ starch test එක කරන්නේ නේද? When we put iodine, when I put iodine, I can't clearly see a color because the leaf is already green color. එතකොට starch නැත්නම් it should be the color of iodine. If there's no starch, it doesn't give a dark blue or purple color. Starch තියෙනවා නම් it will give a dark blue or purple color. But you can't see the colors clearly because the leaf already has a green color. And a starch test එක කරද්දී අපි මොනවාද මුලින්ම කරන්නේ? What are the steps that we do to the leaf before we do the starch test? What do we do to the leaf before we do the starch test? Put the leaf in alcohol and boil it. Would that be the first thing you do? මුලින්ම alcohol වල boil කරා කියලා දැන් අතරම we want to remove the green color නේ. Think about what you want to do. You want to remove the green color. Because when there is the green color, ඔයා දාන මොනවත් හරියට පේන්නේ නැහැ. පේන්නේ නැත්තම් green color එකයි අයින් කරන්න ඕනේ. Green color එක ඇවිල්ලා තියෙන්නේ මොනවා හින්දාද? Green color එක ඇවිල්ලා තියෙන්නේ මොනවා හින්දාද? Yes, chlorophyll hinda. So my ultimate goal should be to remove the chlorophyll. And our first step would be, yes. Then my answers keep your kava answer box, okay? Chat box, okay? First of all, you boil the leaf in water. මේ leaf එක ඔයා මුලින්ම water වල boil කරනවා. එම stand එකක් තියලා you directly heat it and you boil this leaf in water. What is the next step? This is step number 1. What is the next step? කියන්න බලන්න මම කියනකම්. දැන් water වල boil කරන leaf එකට what do we do next? මට දැන් කලින් ආපු answers ටික කියන්න. order එක වරද්දවනේ ඔයගොල්ලන් කිව්වේ. Yes. Now only we put the leaf in alcohol and boil. Then තමයි alcohol වලට දාලා boil කරන්නේ. ඒකත් do I just do it like this? මේ තියෙන alcohol, මේ තියෙන leaf එක. This is alcohol. Yes. Do I just මෙහෙම heat කරනවද? Is this the right way to do this, Tamai? No. Then what is the right way? I got one answer. What is the right way? What is the right way? Yeah. Binuti, yes. What about others? Yes, Ramdira. Yes. Batula, yes. Setumini, yes. We are going to... Yes, Dinugi. We are going to heat it using a water bath. ඒකට double boil කියන වචනේ exam එකේ ලියන්න එපා. I mean, it is double boiling, that is true. Exam එකේ the proper term that we use is heat it using a water bath කියන එක. So water bath එකක් තියෙන එකක මේක immerse කරලා ඒ water එක තමයි අපි heat කරන්නේ. ඒතර ඒ water එකෙන් මෙහෙම heat එක indirectly තමයි leaf එකට යන්නේ. So this is our next step. Boil in alcohol in a water bath. ඊට පස්සේ මේ leaf එක එළියට අරගෙන අපි wash කරලා ගන්න. Now will the leaf be green color? Will the leaf have any green color now? No. Now the leaf will not be green. Hey, alcohol වල ඔක්කොම chlorophyll ටික දිය වෙලා. Now the leaf will be like a very pale color. ඒතර iodine දැම්මම හොඳට color එක පේනවා. දැන් exam එකේ අහනවා ඇයි මේ leaf එක මුලින්ම water වල boil කරන්න කියලා. What is the reason for boiling the leaf in water first? What is the reason for? Okay. අපිට ඕනේ තමයි chlorophyll ටික අයින් කරන්නනේ. Chlorophyll is inside the cells. Cell එක ඇතුලේ chloroplast එක ඇතුලේ තමයි chlorophyll ඉන්නේ. ඒක අයින් කරන්න කලින් අපි මුලින් now cell wall is pretty rigid. නිකන් ගෙදරක තාප්පයක් වගේ. ඒකෙන් මුලින්ම මොකද කරන්නේ? cell wall එක weak කරනවා. We are going to we are not going to exactly destroy. ඒකෙන් destroy කියන වචන use කරන්න එපා, but you can weaken the cell wall. එතකොට cell wall එක ඇතුලෙන් එතකොට යන්න ලේසි. We say to increase permeability. Permeability means ability to go through the cell wall. ඊළඟට අපි මේක alcohol වල boil කරනවා. Why do we boil in alcohol? Why do we boil in alcohol? Yes. දැන් cell wall එක weak වෙලානේ first step එකේදී. Now second step එකේදී leaf එකේ cells ඇතුලේ ඉන්න chlorophyll ඔක්කොම alcohol වල දිය වෙනවා. To remove chlorophyll. Why do we boil using a water bath? අපි directly අල්ලන්න පුළුවන් flame එකට, but why do we use a water bath for this? Why do we use a water bath for this? Yes, because alcohol is highly inflammable. That means even the smallest spark can make it catch fire. That is too risky. That's why we water bath take a heat kara. The chlorophyll alcohol will hinda. Once the leaf comes out, it will no longer be green color. White ma venna hai, but it will be like a very pale color. Ilagaro iodine dana. You put iodine. If they are restarched, that spot will give a dark blue or purple color. Green. So you can clearly see. The places where starch is not present, you cannot see a color change. In a mukha da bit pe color iodine wala brown color ka tamai pain. Okay, that is a color that we can see. In here, part covered with the black glass strip, there is no starch because light avenane, so starch is not produced. Therefore, the color we can see is the color of iodine. That is like a yellow yellow or light brown. Then part covered with colorless glass strips. Yes, colorless glass strips here, you can see a dark blue or purple color. Direct light. Direct light here, yes, dark blue or purple. You can say blackish blue, purple color. It's a very dark purple color. So that's why I'm a dark blue because sometimes it can be confused. Like black color to huru, blue hari, purple hari color. That is it for the second part. That's it for the question. Did you understand this part? Photosynthesis part another. So light when pulang, carbon dioxide when pulang, chlorophyll when pulang. Api antimat hame ke starch test karna akela me mulu process ekama karna. Okay. So let's give the marks. This is 2019. Second question. Mark karaganda. Metana eukarya කියලා 1 mark. Animalia 1 mark. Mammalia 1 mark. Equine automata 1 mark. Mammalia na to mammals කියලා kamak nahi. Here for writing archaea or eukarya 1 mark. Here for writing protista 1 mark. Mollusca, annelida, coelenterata 1 for each 3 marks. Metana light energy, sunlight evage answer 1 mark. Metana to remove stored starch or to remove starch or to de-starch. Evage answer you have gotten 2 marks there. Metana yellow hari brown hari 1 mark. For this 1 mark, this 1 mark. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. How many marks did you get out of 15, Lamai? Okay, Binuti. Akib Tenumi. Batula Uneli Ashini. Dinugi. Senet. Others? Setumini. Batula. Randira. Okay, Randira. Sanutmi. Sorry, Saritma. Okay, Lama. Samaning, if you have 13, 14, 15, it's good. It can put a safe path. So 12, 13 වලට doing තියනවා නම් හොඳට බලාගන්න මොනවද වැරදිච්ච තැන්. So make sure you do a good revision. Okay? So I'm not saying 13 වලට අඩු වෙච්ච ළමයි පාඩම් කළා නැහැ කියන එක නෙමෙයි මම කියන්නේ. පාඩම් කරාට මේ section එක පාඩම් වෙලා නැහැ. Okay? So ඒක හින්දා ඒ section එක පාඩම් කරා. Revise කරා. Okay? Alright, let's move on to the third question. Chemistry. මේක first part එක නම් ලේසි. The symbols of the elements in the second period of the periodic table are given below. Given in the following table in respective order. අපිට order එකත් හරි order එක තමයි දීලා තියෙන කෙලින්ම lithium, beryllium, boron, carbon, nitrogen, oxygen, fluorine, neon. Select the symbol of the element relevant to each of the following statements from the above table. ඒ කියන්නේ දීලා තියෙන elements වලින් තමයි ඔය element එකක් select කරන්න ඕනේ. Maximum first ionization energy. Yes. Neon. First ionization energy කියලා කියන්නේ ළමයි one electron remove කරන්න කොච්චරක් energy දෙන්න ඕනද කියලා. The minimum energy that we need to give to remove one electron from a gaseous atom so that it will become a unipositive gaseous ion. Electron එක අයින් කරාම plus වෙනවනේ. ඒක noble gases වලටත් තියෙනවා. But noble gases වල අයින් කරන්න හරිම අමාරුයි because they're very stable. So ගන්න මතකයි නම් lithium, beryllium, boron, carbon, nitrogen, oxygen, fluorine, neon. This is the second period. කොහොමද first ionization energy vary වෙන්නේ? lithium ගේ තමයි අඩුම. Then second එක කෙනා ඊට වඩා වැඩි beryllium. Third group එක චුට්ටක් අඩු වෙනවා boron. Fourth එක වැඩි වෙනවා carbon. Fifth එක වැඩි වෙනවා nitrogen. Then sixth එක ආයෙ චුට්ටක් අඩු වෙනවා oxygen. Seventh එක fluorine. Eighth එක neon. Then ආයෙ පල්ලෙහාට ගිහිල්ලා we start sodium. So මේ level එකට වඩා චුට්ටක් පල්ලෙහෙන් තමයි sodium, magnesium, the third period goes like that. එතකොට මේ zigzag pattern එකක් තමයි තියෙන්නේ first ionization energy වල. Remember, always the third group and the sixth group go a bit down and then ඊළඟ එකේදී ආයෙ චුට්ටක් උඩට එනවා. First ionization energy. Minimum electronegativity. Minimum electronegativity. Yes, it is lithium. Electronegativity කියන්නේ ළමයි when two atoms are in a bond, what is the capability or what is the ability for that atom to attract the electrons in the bond towards itself? Lithium තමයි අඩුම. Which one has the highest electronegativity here? In these elements, which one has the highest electronegativity? In the given elements. Yes. Neon වලට electronegativity අපි 0 කියලා consider කරනවා because electronegativity කියන්නේ bond එකක තියෙන electrons attract කරගන්න එකනේ. Neon do not form bonds because it is a noble gas. In a maximum electronegativity fluorine. Diamond is one allotropic form of what? Carbon. Now carbon වල allotropic forms දෙකක් තියෙනවානේ. මම ලොකුවට කතා කරන්නේ නැහැ. But simply said, you know, we have crystalline form and amorphous form. මේක අපි ගොඩක් සැරයක් කතා කරලා තියෙනවා, right? Crystalline form එකක් තියෙනවා, amorphous form එකක් තියෙනවා. Crystalline form එකට examples මොනවද ළමයි? What are the examples for crystalline form? Of carbon. Diamond, what else? Graphite. What are the examples for amorphous forms of carbon? Charcoal, coal, soot. Soot is lamp soot dali. So crystalline will they're always crystals, amorphous they're like a powder, not really crystals. exist as diatomic molecules with a triple bond between the atoms. ඒකට මොකක්ද දෙන්නේ? nitrogen. nitrogen කොහොමද අපි draw කරන්නේ? nitrogen ගේ electronic configuration එක 2 5. If you take one nitrogen, there is 1 2 3 4 5. If you take another nitrogen, it's the same. Then කොහොමද bonds හැදෙන්නේ? මෙයාගේ ඉලෙක්ට්‍රෝන එකයි මෙයාගේ ඉලෙක්ට්‍රෝන එකයි බොන්ඩ් වෙනවා, මෙයාගේ ඉලෙක්ට්‍රෝන එකයි මෙයාගේ ඉලෙක්ට්‍රෝන එකයි බොන්ඩ් වෙනවා, මේ ඉලෙක්ට්‍රෝන එකයි මේ ඉලෙක්ට්‍රෝන එකයි බොන්ඩ් වෙනවා. So it will be 3 bonds. And මෙයාගේ එක lone pair එකක් ඉතුරුයි, මෙයාගේත් එක lone pair එකක් ඉතුරුයි. So like this. Okay. So nitrogen is the atom that makes a diatomic molecule කියන්නේ atoms දෙකක් එකතු වෙලා molecule එකක් හදනවා where there is a triple bond. Exist as diatomic molecules as well as triatomic molecules in the atmosphere. මේකට answer එක ළමයි ozone. තම biosphere recording කෙව්වේ නැහැනේ, so ඒක බැලුවට පස්සේ it will be clear. Sorry, oxygen. Answer is oxygen. Why? Because oxygen atmosphere ke inna ekak oxygen gas එක විදියට, which is O2. We know that is what we use for respiration and what is released during photosynthesis. That is the oxygen gas, O2. A little above in the atmosphere, there's the ozone layer. Ozone කියලා කියන්නේ O3. And that is also a gas. So ozone layer ke තනිකර මේ O3 gas molecules වලින් තමයි මුළු layer එකම හැදිලා තියෙන්නේ. Okay, so answer here should be oxygen. Write the formula of the compound formed by the combination of lithium and oxygen. What is the answer? Yeah. Others? Lithium and oxygen. What about others? Hamalima periodic table likhe. Yes. First group ke valency 1, second group ke valency 2, third group ke valency 3, fourth group ke hamogema valency 4, fifth group ke katte again 3, 2, 1 and 0. 1, 2, 3, 4, 3, 2, 1, 0. Lithium ke valency 1, oxygen ke valency 2. So cross sunama lithium to 2 enawa, oxygen to 1 enawa. That would be Li2 compound formed by the combination of lithium and oxygen. මෙතන මොකද lithium ගේ cation එක as you think? As you think නෙමෙයි, as you know this already. Lithium has the valency of 1 නේ. එයාගේ valence එක 1 නේ. ඒ කියන්නේ හැම වෙලෙම එක electron එකයි අයින් කරන්නේ. කොහොමද ළමයි lithium ගේ electronic configuration එක තියෙන්නේ 2 1. So it will always remove only 1 electron. Enang it will form lithium plus. Oxygen ke valency 2 means that 2 electrons ganna. Valency 2 කියන්නේ 2 electrons ganna. එහෙම නැත්නම් oxygen ke configuration එක 2 6. In order to become stable, in order to become 2 8, එයා electrons දෙකක් ගන්න ඕනේ. දෙකක් ගත්තාම එයා O2 minus වෙනවා. So answer is lithium plus and O2 minus. So lithium plus, මොකද අපි කරන්නේ underline නේ. Lithium plus and O2 minus. Hydrocarbons recordings එක බැලුවද ළමයි? Did anybody watch the hydrocarbons recordings? Did you all draw this monomer? Make a monomer කන්දද? Okay. Hydrocarbons will the recording would be a little more than matname would be roughly about a week ne. So I think that should be enough time to watch the recording. මොකද අද හෙට දවස් දෙකේ බයොස්ෆියර් රෙකෝඩින්ග්ස් උත් එවනවා. එතකොට ඒ ටිකත් බලන්න. Carbon and fluorine in a monomer එක මොකක්ද? tetrafluoroethene. So you know ethene is like this නේ. Ethene is like this. Ethene molecules එකතු වෙලා හදනවා polyethene නැත්නම් polythene. So polythene ගත්තොත් එහෙම this is the monomer. Then ඒකෙන් හදනවා polythene කියන polymer එක. So මෙයාගේ double bond එකේ එකක් break කරලා මේ පැත්තට හදනවා. මේකෙන් එකක් break වෙලා මේ පැත්තට හැදෙනවා. So I'm not going to talk too much about it because this was in the recording and you must have recently watched it. So you have this fresh in your memory. So this is the repeating unit. මේ වගේ units ගොඩාක් repeat වෙලා තමයි polythene හැදෙන්නේ. So polymer එක පෙන්නද්දී මට මේ වගේ දේවල් මේ වගේ ගොඩාක් ඇඳින්න බෑනේ. So polymer එක represent කරන්නේ මම repeating unit එකටම I put a bracket and I put n. This will be repeated as many times. Okay. May I will polythene. Now this is polythene. How many tibba polymers? අපි ඉගෙන ගත්තා. Remember there was a monomer like this. Ethene වගේම තමයි, but there are 4 hydrogens නෙවෙයි, there are actually 3 hydrogens. එක hydrogenයක් වෙනුවට ඇවිල්ලා chlorine එක්කෙනෙක්. What is this molecule called? What is this molecule called? Chlorofluoroethene නෙවෙයි, fluorine නෑනේ. It's chloroethene. Okay. This is chloroethene and this is the monomer. මෙයා ගොඩක් එකතු වෙලා තමයි polymer එකක් හැදෙනවා PVC. polyvinyl chloride කියන polymer එක. So කොහොමද හැදෙන්නේ? මේ double bond එක කැඩෙනවා. කැඩිලා මෙහෙම දෙපැත්තට bonds දෙකක් හැදිලා ඒ වගේ units ගොඩාක් එකතු වෙලා තමයි හැදෙන්නේ. Like this. So මෙතන double bond එක එකක් හැදිලා so there will only be one bond. And මෙහෙම දෙපැත්තට හැදෙනවා. So මේ වගේ ගොඩාක් repeat වෙලා තමයි polymer එක හැදෙන්නේ. So when we draw the polymer, we draw like this. Repeating unit එකටම we put a bracket and we put n. That means this is repeated as many times. This polymer is called what? මේ polymer එකට අපි මොකද්ද කියන්නේ ළමයි? Polychloroethene, or we call it polyvinyl chloride. Polyvinyl chloride is a term that we use the most. That is PVC. You know PVC pipe පාරම්මේව තියෙන්නේ? So that is PVC. This is that polymer. Then there's another polymer. Do you remember what that is? Recording එකේ තිබ්බනේ තව polymer එකක්. So carbon අර ethene වල තියෙන hydrogen හතරක්නේ. Instead of 4 hydrogens, here there are 4 fluorines. This is actually what they're asking in the question. Eh? What is this molecule called? මෙතන fluorine හතරක් තියෙනවා. So we call it, හතරක් means tetra. So we call it tetrafluoroethene. Ethene වලම hydrogen. Now ethene is this no? මෙතන hydrogen හතර වෙනුවට we have attached 4 fluorines. So tetrafluoroethene. So මෙයාම monomer එක විදියට අරගෙන polymers හදනවා. So what happens? Double bond එක කැඩිලා දෙපැත්තට bonds දෙකක් හැදෙනවා. ඒ වගේම ගොඩක් join වෙනවා. So like this. Hydrocarbons recording එක බලපු කට්ටිය did you all understand everything in that? ඒක ගොඩක් රෑ වෙලා මේ මහන්සියෙන් කරේ so I was a bit worried whether if everything is clear in that recording. Balpu katte, is it all good? Was everything clear in the recording? එකයි මම සමහර තැන්වල කැමරා එක off කරගෙනත් හිටියේ. But since the recording was urgent, I had to send it. That's why I recorded it even then. But if you have any questions or any concerns, any clarifications you need, please let me know. Kumari, this is the repeating unit and this is the polymer of that. මේ polymer එකට අපි කියනවා polymer එකට fluoroethene වලින් හැදිච්ච polymer එක. So we call it polytetrafluoroethene. Tetrafluoroethene වලින් හැදිච්ච එක we call it polytetrafluoroethene. In simple terms, we call it Teflon. Teflon pans එහෙම තියෙන්නේ. So that is Teflon. So these are the main three polymers that we learned. Okay. So question එක අහන්නේ, write the structure of the simplest monomer molecule which can polymerize and contain the. They are talking about tetrafluoroethene. They are talking about tetrafluoroethene. Okay, Lama, is the third A part clear to you before we move on to the next part? Thumbs up, please. Okay, we'll move on to the B part. The following flow chart shows a series of reactions carried out starting with calcium carbonate. Okay. අපි මේක චුට්ටක් පැත්තෙන් sketch කරමු. There's no space. Let me move this to this side. Calcium carbonate solid එක. That is what we have here. Calcium carbonate solid. මම මේ ඔක්කොම symbols වලින් අදලා බලන්නම් පැත්තෙන්. So we have calcium carbonate solid. And I'm going to heat it. Heat symbol එක triangle එකනේ. Then U කියලා solid එකකුයි V කියලා gas එකකුයි හැදෙනවා. When you heat calcium carbonate, what are produced? අර කමතක කරන්නකෝ. As we know, what are produced when you heat calcium carbonate? Decomposition reaction again. Chemical reactions are මතක තියාගන්න ළමයි, because you are expected to memorize. ඒව නම් කටපාඩම් කරන්න වෙනවා. Chemical combination reactions හතරක් තිබ්බා. Chemical decomposition reactions හතරක් තිබ්බා. Then single displacement ඒවත් හතරක්ද කොහෙද තිබ්බා. Double displacement I think again 3 or 4 තිබ්බා. ඊට පස්සේ metals එයා එක react කරන විදිය, metals water එක react කරන විදිය, metals dilute acids එක react කරන විදිය ඒ ටිකත් බලා ගන්න ඕනේ. Then extraction of ion වල reaction set එකක් තිබ්බා. What else? I'm going to quit the reactions තිබ්බේ. Preparation of hydrogen gas, there was one reaction. Preparation of oxygen gas, there were 4 reactions. ඒ ඔක්කොම දැනගන්න ඕනේ නැහැ. Then preparation of carbon dioxide also, there was one reaction. I am a chemical reaction set එකක්ම තිබ්බා මේ electrochemistry වල. Okay. Anyways, calcium carbonate decompose වුණාම when you heat, it will be a decomposition reaction. It will form calcium oxide and carbon dioxide. මෙතන කියනවා නේද U කියලා solid එකක් කියලා and V කියලා gas එකක්. So U කියලා solid එකක් means it should be this name. මෙයානේ solid එක. V කියලා gas එකක් means it has to be carbon dioxide. So U is calcium oxide, V is carbon dioxide. U කියන එකේනට water දැම්මම W කියලා solid එකක් එනවා. W කියන solid එකට excess water දාලා stir කරලා filter කරාම X කියලා clear solution එකක් එනවා. බලන්න X කියන clear solution එකට when we bubble V, you know V is carbon dioxide, right? So X කියන එක්කෙනාට මම carbon dioxide bubble කරාම calcium carbonate හැදෙනවා. What is X then? What is X then? Remember lamai, when we were testing for carbon dioxide, අපි carbon dioxide gas එක යවුවා lime water වලින්. Lime water වලින් carbon dioxide යවුවාම it turned milky color. I told you it turns milky color because calcium carbonate is formed. When there is a lime water sample, you know lime water කියන්නේ calcium hydroxide. Calcium hydroxide වතුර වල දිය කරාම that is AQ. මෙයාට මම carbon dioxide යවුවා මොකද වෙන්නේ? තනින් තන මෙහෙම milky suspend වෙලා තියෙනවා milky color එකට particles. That's why this entire thing looks milky color. That is because calcium hydroxide and carbon dioxide react, forming calcium carbonate. Calcium carbonate and water හැදෙනවා. But calcium carbonate හැදෙනවා. Now this is white color. That is why this gives a milky color. එහෙනම් මොකකට carbon dioxide යවුවාමද calcium carbonate හැදෙන්නේ? Calcium hydroxide වලට. Therefore, here X should be calcium hydroxide. Calcium hydroxide solution AQ, which is, in other words, we call lime water. මෙතන ළමයි W කියලා කියන්නේ calcium hydroxide වලට water දාලා filter කරාමනේ calcium hydroxide හැදෙන්නේ. W වලට water දාලා filter කරාමනේ. So actually W is calcium hydroxide solid. We call it slaked lime. ඒ experiment එකේ ඔයාලට මතකයි නම් අපි slaked lime ටිකක් අරගෙන, that means calcium hydroxide solid අරගෙන වතුර දිය කළ තමයි lime water හදාගත්තේ. එහෙනම් now we know what is X and we know what is W. Okay. So අපි U අහනවා, U is calcium oxide, W අහනවා, W is calcium hydroxide. ඒක නිකන් ලියන්න බෑ, chemical formula එකයි ලියන්න ඕනේ. You can't write in words. Calcium oxide වචනෙන් ලියුවොත් that is quicklime. Calcium hydroxide solid එක අපි වචනෙන් ලියුවොත් that is slaked lime. So වචනේ ලියලා හරියන්නේ නැහැ, we have to write the chemical formula. Did you all understand this question a bit? Okay. Delete one word printed in bold so that the correct idea is expressed. Solid U is acidic or basic. Solid U කියන්නේ මෙයා. බලන්න මෙයාට වතුර දැම්මම හැදෙන calcium hydroxide නේද? Calcium hydroxide වල තියෙනවා OH minus. OH minus that it is a base. If calcium hydroxide has OH minus and it is a base, calcium oxide should also be basic. එයාට වතුර ටිකක් විතරක් දැම්මම එයා OH minus තියෙන එක්කෙනෙක්ව හදනවනේ. OH minus තියෙන එක්කෙනෙක් කියන්නේ it's a base. ඒතර එයාව හදන්න නම් මෙයාගේ basic properties තියෙන්න ඕනේ. So this is also basic. බලන්න ළමයි මේක work out කරන විදිය question එක right? Answers directly අපි ඉගෙන ගත්ත දේවල් they won't like straightforwardly ask it. They will twist it and ask. Okay? You know OH minus means it's a base and there is OH minus here and here both. That means they are both bases. Base එකක් හැදෙන්නේ base එකක ඉන්නේ. So calcium oxide is also a base because we just added water to calcium oxide, right? So calcium oxide is basic. එහෙනම් acidic කියන වචනේ මම කපනවා. The bond in the solid U is ionic or covalent. Solid U කියන්නේ calcium oxide. But calcium is a metal. Oxygen is a non-metal. When metals and non-metals react, it is an ionic compound for sure. So covalent කියන එක කැපුවා. The pH value of an aqueous solution of gas V. ළමයි gas V කියන්නේ carbon dioxide නේ. Carbon dioxide කියන්නේ මේක biosphere එකේ තමයි කතා කරන්නේ. But for now I will tell you the answer. Later biosphere එක ඉගෙන ගත්තාම ඒකත් එක්ක compare කරන්න පුළුවන්. Carbon dioxide is a gas that is acidic. Carbon dioxide can acidic gas එක. So they are saying pH value of an aqueous solution of gas V. So gas V, we are going to dissolve in water. Gas V is carbon dioxide. So carbon dioxide is an acidic gas. When you dissolve it in water, that solution will be acidic. Acidic කියන්නේ, is it higher than 7 or lower than 7? Acidic means lower. Acid එකක් කියන්නේ pH හැමලෙම 7 වලට වඩා අඩුයි. So this is lower. එහෙනම් higher කියන එක කපනවා. Okay. What can be seen when excess of gas V is bubbled through the milky solution? Milky solution එක හැදුන මං කිව්වේ lime water, which is calcium hydroxide AQ. මේකේ balance reaction එක නැතත් කමක් නැහැ. ඒකට මම carbon dioxide pass කරාම calcium carbonate හැදුනා. And calcium carbonate is white, therefore I was able to see a milky colored solution. What can be seen when excess of the gas V is bubbled through the milky solution? මේකට මම තවත් carbon dioxide යවනවා. දැන් මොකක් වෙයිද? Now what will happen? මේකත් අපි කතා කරානේ. Lime water වලට carbon dioxide gas යවුවාම it becomes a milky color solution because of calcium carbonate. එයාට තවත් carbon dioxide යවුවාම මොකද වෙන්නේ? It turns colorless. Colorless වෙන්නේ ඇයි? Because calcium bicarbonate is produced. Calcium bicarbonate කියන්නේ colorless එකනේ. So you know calcium වල valency 2, bicarbonate වල valency 1, ඒකයි cross වුණාම මෙහෙම ආවේ. එහෙනම් මෙතන අපෙන් compound එක අහන්නේ what can be seen. What you can see is that the solution becomes colorless. Up to this point, is everything in this question clear? Give me a thumbs up. Others? Dinugi, do you have a question? If you have any questions, ask. Huh? My marks then now. May occur 1 for each hambela neon 1, lithium 1, carbon 1, nitrogen 1, oxygen 1, 1 for each. Then for writing lithium oxide, ekarat 1 mark. For selecting lithium plus 1 mark or 2 minus 1 mark. Then make a monomer andinawata, you have gotten 1 mark. Maybe විදියටම ලියලා තියෙනවා විතරයි 1 mark. මොකක් හරි එකක් හරි වැරදි නම් it's 0. මෙතන calcium oxide කියලා ලිව්වා නම් 1 mark. මෙතන calcium hydroxide කියලා ලිව්වා නම් 1 mark. මේ solid එක තිබ්බේ නැතත් කමක් නැහැ. Solid කියලා දැම්මේ නැතත් කමක් නැහැ. Just calcium hydroxide කියලා ලිව්වා නම් ඇති. මෙතන acidic කියන එක strike off කළා basic answer එක ඉතුරු කරා නම් 1 mark. Covalent strike off කළා ionic ඉතුරු කරා නම් 1 mark. මේකට 1 mark. මේකටත් 1 mark. ඒක නම් exam එකේ free marks දීලා තියෙනවා but ඕගොල්ලන් හරියට mark කරගන්න. It's pretty clear I think. Yeah. So 1 2 3 4 5 6 7 8 9 10 11 12 13 14. 5 6 7 8 9 10 11 12 13 14 15. How many marks did you get out of 15? Okay, Akib, Tenumi, Ashini, Binuti, Saritma, Batula, Oneli, Disal, Dinugi. So තුන්දෙනෙක් ඉන්නවා. Others? Saritma, Fatima. Okay. Let's move on to the next question. But before that, give me 2 minutes. I'll be back. इतका होगा ना वहाँ recorrections थे ना 1 मिनट करेगा. Okay, Lamai. So now we'll move on to the fourth question. Let me get the question to the whiteboard. We have current electricity first. So a teacher provided the following materials and equipment to students: a nichrome wire coil, 4 dry cells, ammeter, switch, voltmeter, rheostat. Okay. Incomplete circuit diagram. By what name is the way the dry cells are connected in the circuit node? කොහොමද මේ dry cells connect කරලා තියෙන්නේ? Hmm? Series or parallel? Series. In series or series combination. Series කියන වචන තියෙනවා නම් you get marks. Excuse me. Draw in the circuit diagram the standard symbol of the equipment that should be connected between the terminals X and Y. What should we connect between X and Y? What should we connect between X and Y? Ammeter. මොකද ammeter එකක් නේ දීලා තියෙන්නේ. එහෙනම් ammeter එක කොහොමද අපි draw කරන්නේ? මෙහෙම A කියන එක දාලා circle එකක් දාන්න. එහෙම ඇඳලා තියෙන්න ඕනේ. Why is a rheostat connected to this circuit? Why do we connect? Yes. Why do we connect a rheostat, lamai? Yes, to change the current or to control the current or to control the voltage, potential difference, anything like that. State a condition that would result if the switch is kept closed for a long time after completing the circuit correctly. What will happen? Yes. So, what would happen if the switch is kept closed for a long time after completing the circuit correctly? Okay, yes, increasing the resistance. Hari. ඔගලන්ට හොඳට කියන observation ගන්න පුළුවන්. Resistance is something you cannot see. Resistance is correct, but it's not something that you can see. What would be an observation that you can get here? Observation කියලා අහුවොත් මෙතන state a condition නැතුව what if they ask observation? What is the observation you can get if the switch is kept closed for a long time after completing the circuit? Yes. This circuit will heat up or the temperature will increase. That is an observation. මෙතන observation කියලා නැති හින්දා you can write increasing the temperature or heating the coil, heating of the coil or heating of the circuit, or you can write increase in resistance. ඕන එකක් ලියන්න පුළුවන් ඒ වගේ. Given here is a graph of V against I. V means voltage, I is current. What is the physical quantity represented by the gradient? That is easy, no? That is resistance. V versus I, voltage versus current එකක gradient එකෙන් දෙන්නේ resistance. Okay. Resistance අතෝ R කියලා ලිව්වත් කමක් නැහැ. Let's move on to the next part. Let me get the question to the whiteboard. The following activity was done by a group of students to find the center of gravity of a metal statue. The head and 2 feet of the statue were placed on the smooth pans of 2 identical balances X and Y, kept on a horizontal flow as shown in the figure. Reading in balance X was 250, reading in balance Y was 150. What are the forces acting with regard to the equilibrium of the statue? What are the forces acting here, Lamai? Yes. එකක් gravitational force කියන්නේ weight. Weight කියලා ලිව්වත් කමක් නැහැ, W කියලා ලිව්වත් කමක් නැහැ. What else? R කියන්නේ මොකද්ද ළමයි? You can see R is the perpendicular reaction force from this scale නේ. ඕනම surfaces දෙකක් when they are in contact with each other, there is a perpendicular reaction force acting perpendicularly, right? So perpendicular reaction force at A කියන්නේ එක R. Then perpendicular reaction force at B. එයාගේ reaction කියන්නේ force එකේ නම S. නැත්නම් නිකම් perpendicular reaction force කියලා perpendicular reaction force R, perpendicular reaction force S. එහෙම ලිව්වත් කමක් නැහැ. What is the reaction R through the point A and the reaction S through the point B? Usually lamai when you keep something on a scale, okay, scale එක දෙන reading එක is equal to the perpendicular reaction force at that place. Then what would be the perpendicular reaction R? 250 newtons. And S? 150 newtons. Yes. Then what is the weight W of the statue? What is the weight W of the statue? You know, when an object is in equilibrium under three parallel forces, එක පැත්තක තියෙන force එක equal අනෙක් දෙන්නගේ force එක add කරාම එන value එක. And a W should be equal to R plus S. R is 250, S is 150. So 250 plus W should be equal to R plus S. 250 newtons plus 150 newtons. That would be 400 newtons. So now weight take 400 newtons. The distance from A to the center of gravity of the statue is D. Write the moment of weight of the statue around point A. එකනේ point A තමයි fixed point එක. A point එකේ ඉඳන් W වල moment එක තමයි කියන්න ඕන. What is the answer here? Now we know moment equals force multiplied by perpendicular distance of force from the axis of rotation. If you take W වල force එක W. That is weight, no? Perpendicular reaction force එක දැන් ඔයගොල්ලන් දීලා තියෙනවා D කියලා. See? The force is D. So the distance is D. Then W වල value එක අපි already දන්නවනේ 400 කියලා. So W වලට I write 400 and perpendicular distance is D. So මට ආවේ algebraic term එකක් විතර ලියන්න පුළුවන් 400D කියලා. WD කියලා ලියලා තිබ්බ ලකුණු දීලා තිබ්බද? Let me check. Yeah. මේක මෙහෙම නිකන් W multiplied by D කියලා WD කියලා ලියලා තිබ්බත් it's okay. Lama, is this 400D or WD moment, is it a clockwise moment or an anti-clockwise moment? Clockwise moment or anti-clockwise moment? මේ statue එක fix කරලා තියෙන A වලින්නේ. එතකොට මේ W කියන force එක හින්දා එයා clockwise පැත්තට rotate වෙන්නේ anti-clockwise පැත්තට. මෙහෙම නේද rotate වෙන්නේ මේ? That is clockwise. That is clockwise. හරි ඒක අහලා නැහැ, I'm just asking. What is the moment of S around A? A තියන්නේ මෙතන. A වලනේ fix කරලා තියෙන්නේ. So S වල moment එක තමයි අපෙන් අහන්නේ. Again, we know moment is equal to FD. S වල S ගත්තොත් එම ළමයි S වල force එක අපි already දන්නවා, that is 150 newtons. මෙතන බලන්න fixed point එක A නේ. A ඉඳන් S වලට distance එකත් අපි දන්නවා. It's 2 meters. They have given us that. So 150 multiplied by 2 is 300 Newton meters. So එහෙනම් මෙතන answer එක මොකක්ද? 300 Newton meters. ඔයාට ඕන චුට්ටක් මෙහෙම පෙන්වන්න පුළුවන් 150 Newton multiplied by 2 meters. එහෙම දැම්මේ නැතත් කමක් නැහැ. So since A is the fixed point, is S a clockwise moment or an anti-clockwise moment? Is S a clockwise moment or an anti-clockwise moment? Yes. බලන්න S හින්දා rotation එක තියෙන්නේ මෙහෙම නේද? That is anti-clockwise direction, right? මෙහෙමනේ තියෙන්නේ මේ. Anti-clockwise. So S is anti-clockwise, W is clockwise. The anti-clockwise moment of S around point A is equal to the clockwise moment of W around point A. ඒක ඒගොල්ලන් දෙන්න ඕනෙත් නැහැ. අපි දන්නවා equilibrium නම් කොහොමත් equilibrium එකක නම් anti-clockwise moments equals clockwise moment. ඒක දුන්නේ නැතත් අපි දන්නවා. So anti-clockwise moment එක මොකක්ද? S නේ, that is 300 Newton meters. Clockwise moment එක W, ඒක WD, that means 400 multiplied by D. මම මෙතන මෙයාගේ Newton meters කියන unit එක දැම්මේ නැහැ පැටලෙයි කියලා. So I will change the sides. 400D is equal to 300. Divide both sides by 400. So 400 cancel වෙලා D is equal to මේ zeros දෙකට මේ zeros cancel වෙලා we get 3/4 meters. In other words, that is 0.75 meters. Marking scheme එකේ step එකට ලකුණු දීලා තියෙනවා. ඒ කියන්නේ ගොඩක් ලොකු steps ලියන්න ඕනේ නැහැ. මෙන්න මේ step එක කියන්නේ 300 divided by 400 කියලා ලිව්වත් ඇති. So මෙන්න මේ steps ටික ලිව්වා නම් ඇති. Because you anyways don't have much space now. So මේ step එක ලිව්වා නම් ඇති. Answer එක 3/4 meters කියලා ලිව්වත් කමක් නැහැ. 0.75 meters කියලා ලිව්වත් කමක් නැහැ. So ඒකේ ලකුණු දීලා තියෙන්නේ මේ step එකටයි final answer එකටයි. Up to that point, is it clear, Lamai? Any questions? If it's clear, give me a thumbs up. Okay. Okay, I'll tell you how marks are given here. Series කියලා ලිව්වට 1 mark. Ammeter එක හරියට ඇන්දට 1 mark. To change the current or voltage or to control the current or voltage, 1 mark. Increase in the temperature, heating circuit, increase in resistance, whatever, 1 mark. For writing resistance or even R, it's okay, 1 mark. Then here, it leaves 1 mark, perpendicular reaction force R reaction, what a 1 mark. මෙතන එකක් W, R, S කියලා ලිව්වත් ඇති, right? W is this force, R is this force, and S is this force. Just writing the 3 forces is okay. 250 newtons, 1 mark. 150 newtons, 1 mark. 400 newtons, 1 mark. Make it a 1 mark. Make it a 2 marks. Here for this step, 1 mark. For the answer, 1 mark. That's too much, no? One, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, or 15. Okay. How many marks did you get out of 15? Okay, Bhuti, Tenumi, Akib, Setumini, Uneli, Fatima, Yashini, Visal, Randira, others, Saritma, okay. Others also let me know. Senate, okay. Let's move on to the 5th question. That's essay part, ne? Part B. Answer only 3 questions from the questions 5, 6, 7. That's not relevant to us. Okay. Figure 1 represents a sketchy line diagram which shows the human heart. Name the blood vessels A, B, C, and D. What is A, Lamai? A, what is A? Others, what is A? What did you write for A? Yes. A is superior vena cava. When A, B, C, D, nene. Then B is this. What is B? Others, what is B? Yes, B is aorta. Then what is C? C enne me right ventricle එකෙන් නේ. Right ventricle එකේ මෙහෙම දෙපැත්තට branch වෙලා. Yes, C is pulmonary artery. Then what is D? D කියන්නේ මෙහෙම දෙපැත්තෙන් එන්නේ. ඇවිලා left atrium එකට එන්නේ, so that is pulmonary veins. D is pulmonary veins. When comparing with the composition of the blood flowing through D, what is the main difference in the composition of blood flowing through C? C කියන්නේ pulmonary artery. D කියන්නේ pulmonary vein. What is the difference? Pulmonary artery එකේ pulmonary vein එකේ යන blood වල මොකද difference එක? අහන්නේ අපේ අතරම main difference of the composition of blood flowing through C. C ගැන අපෙන් අහන්නේ. C කියන්නේ pulmonary artery. Pulmonary artery contains deoxygenated blood. What on C and D compare කරලා ලියන්න පුළුවන්. C contains deoxygenated blood. D contains oxygenated blood. එහෙම compare කරන්නත් පුළුවන්. නැත්නම් අහන්නේ composition of blood through C නේ. So compared to D, C වල මොකක්ද difference එක කියලා. So you can write C contains deoxygenated blood. Oxygen concentration in blood is low or something like that. What is a valve named E? E is here. What is this? Yes. Tricuspid නෙවෙයි. Tricuspid තියෙන මේ පැත්ත. This is bicuspid. E is bicuspid valve. Describe briefly how the characteristic lub and up sounds heard during the heartbeat are generated. Lub හැදෙන්නේ කොහොමද? Lub. Lub and up කොහොමද හැදෙන්නේ කියන අහන්නේ lub කොහොමද හැදෙන්නේ? Yes. Yes. ළමයි right atrium එකෙන් right ventricle එකට deoxygenated blood යද්දී ඒ වෙලාවෙම කලින් cycle එකේ oxygenated blood ටික left atrium එකේ ඉඳන් left ventricle එකට යනවා. අපි හිතන්න එක cycle එකක් ඉවර වෙලා දැන් මෙහෙම ඇවිල්ලා මෙතනින් ඇවිල්ලා මෙහෙම ගිහිල්ලා ආයෙ මෙහෙම ඇවිල්ලා මෙහෙම ගිහිල්ලා ඒ cycle එක ඉවරනාට පස්සේ අලුත් කට්ටිය එනවා කියන්නේ එහෙම නෙවෙයි. එක cycle එකක් ගිහලා ඒක මැදදී යද්දී තව cycle එකක් එනවා. So මෙතන deoxygenated blood right atrium in the right ventricle යද්දී මෙතන oxygenated blood left atrium එකේ නම් left ventricle එකට යනවා. They both happen simultaneously. Once they are done flowing, here the tricuspid valve and the bicuspid valve both will close. A tricuspid valve එකයි bicuspid valve එකයි close වෙන sound එකට තමයි අපි කියන්නේ lub sound කියලා. So lub sound is generated when tricuspid and bicuspid valves close. What about the dub sound? I already got one answer. Others, how do we get the dup sound? Yes, others? And then afterwards, what happens is that මෙතන deoxygenated blood ටික left right ventricle එකේ ඉඳන් pulmonary artery එකට යනවා. ඒ අතරේ oxygenated blood ටික left ventricle එකේ ඉඳන් aorta එක through යනවා. ඒ දෙන්නම ගිහිලා ඉවරුනහම මෙතන තියෙන semilunar valve එකයි මෙතන තියෙන semilunar valve එකයි එකම වෙලාවට තමයි close වෙන්නේ. That is the dup sound. Semilunar valves දෙකක් තියෙනවනේ නේ? When semilunar valves close. Then fifth Roman number. Figure 2 is this. Again, ECG එකක් නේ? Indicates the potential differences, okay? Which stage of the cardiac cycle is denoted by T? මම මුලින්ම කියන්නේ P මොකද්ද? What is P? What is P? What is P ළමයි? Okay, others? What is Q? Q කියන්නේ Q තනියෙන් නෙවෙයිනේ, it's QRS. What is QRS? Okay. Yes. Here P කියන්නේ atrial contraction. That means atria දෙක contract වෙන එක. P means atrial contraction. In other words, we also call it diastole. Q කියන්නේ මොකද්ද? As you said, Q තනියෙන් නෙවෙයි ඉන්නේ, QRS කියන්නේ තනිකර එක signal එකක්. So Q තනියෙන් නෙවෙයි, QRS together, that is a ventricular contraction. What is the word that we call ventricular contraction? මට chat box එකේ තාවා systole. Finally, what is T? T එක මම මෙතනම ලියන්නම්. What is T? T is intervening. Or we can also call it complete cardiac diastole. Intervening, complete cardiac diastole, ඒ වගේ වචනයක් දැම්මම හරි. නැත්නම් atrial and ventricular relaxation. එහෙම ලිව්වත් කමක් නැහැ. Atrial and ventricular relaxation. One of these ලිවලා නම් ඇති. For T. අහන්න අපෙන් T නේ. Then Roman number 6. Heart muscles are weakened by the blockage of blood vessels supplying blood to them by blood clots. What is the name of this ailing condition? Blood clots in the venom, that is thrombosis. Atherosclerosis නෙවෙයි ළමයි. Atherosclerosis වල වෙන්නේ blood vessels වල මේ vessel එකේ size එක අඩු වෙනවා because cholesterol and other fat will come and deposit in the vessel. If you take a blood vessel like this, make it blood යන්නේ, but fat and cholesterol and all of that will deposit in the blood vessels. Then the size of the vessels becomes like smaller. So there is no space for blood to push through. හැබැයි කොහමරි blood යවන්නත් උන හැබැයි ඉඩක් නැහැනේ. එතකොට මොකද කරන්නේ? heart එක තවත් වැඩියෙන් pump කරනවා, pressure එක වැඩියෙන් දෙනවා to try and send the blood through these vessels. That is what we call atherosclerosis. එතකොට අපේ heart එක රිදෙනවා. Extra pressure එකක් exert කරනවානේ. Then we call it angina or chest pain. Okay. Let's move on to the next question, B. B is about inheritance. The number of chromosomes in a somatic body cell of a mammalian animal species X is 40. අපේ තියෙන්නේ කීයද 46. දැන් මේ කතා කරන animal species ට තියෙන්නේ 40. What is the number of chromosomes contained in the daughter cells produced by meiotic division of a gamete mother cell of X? So X ගේ chromosomes 40 නම් gamete mother cell එකක් meiosis වලින් divide වුණාම කොච්චර chromosomes යනවද daughter cell ට? You know, one of the main features of meiosis is that the number of chromosomes become half. Neither number of chromosomes become half. If the species have 40 chromosomes, meiosis valadi that become half and will have only 20 chromosomes. Name a daughter cell type formed by the meiotic division of gamete mother cells of X. Daughter cell type එකක් කියන්නේ මොකක්ද? What is the answer you wrote here? Basically, they are asking මෙහෙම 20 chromosomes තියෙන cell එකකට example එකක් නේ. So meiosis වලින් හැදෙන්නේ මොනවාද? In males it would be the sperm. If it's females it would be the egg or the ovum. එහෙනම් මේකේ answer එක ඒක තමයි. Either sperm or you can write ovum or you can even write egg cell. එතකොට ඔය තුනේ එකක් එකයිනේ අපෙන් අහන්නේ නේ එකක් ලියලා තියෙනවා you get marks. How does a daughter cell formed by the mitotic division differ from a cell formed by meiotic division? Mitotic division වල මොකද්ද වෙනස meiotic එක compare කරද්දී? Mitotic වල අහන්නේ ආ? Yes. Meiotic එක compare කරාම mitotic වල වෙනස මොකද්ද? Number of chromosomes in the daughter cells is equal to the number of chromosomes in mother cell. Or you can say daughter cells are identical to the mother cell or something that goes along the lines. If you have different answers, let me know. Then C. The garden pea plant seeds have two shapes, round and wrinkled. The gene giving rise to round seeds is capital R, while the gene giving rise to wrinkled seed is simple r. The genotype of the plant with dominant homozygous genes for this seed shape is capital R, capital R. Write the genotype for each of the following plant with regard to seed shape. Genotype එකන genetic composition එක. අපි දන්නවා dominant homozygous එක්කෙනා capital R, capital R. අපෙන් අහන්නේ recessive homozygous. What would that be? Recessive කියන්නේ simple R නේ. Recessive homozygous means එකම එක්කෙනා repeat වෙලා. And a recessive homozygous means simple R, simple R. Plant with heterozygous genes. Heterozygous කියන්නේ genes දෙන්න දෙකක්. So එහෙනම් capital R, simple R. When a pure breeding garden pea plant with round seeds, pure breeding garden pea plant with round seeds කියන්නේ capital R, capital R. Was crossed with a pure breeding garden pea plant with wrinkled seeds. That means simple R, simple R. All the F1 generation had round seeds. So make a P generation එක නම් parental generation. අපි බලමු gametes will just draw a cross. capital R ගේ gamete කොහොමද තියෙන්නේ capital R විතරයි නේ කියලා මම එකක් ලියුවා නම් ඇති. Simple R ගේ හැමෝම simple R හින්දා I'm just writing one. So everybody in the F1 generation will be capital R, simple R. Capital R, simple R කියන්නේ round seeds. එතන ඒගොල්ලන් කියන එක හරිනේ. They are saying all the F1 generation plants had round seeds. ඇත්තනේ ඉතින් capital R, simple R means round seeds. In the F2 generation obtained by crossing two plants in the F1 generation. F1 generation එකේ දෙන්නේක cross කරලා F2 generation එක ගන්නවා. Ratio of plants with round seeds to wrinkle seeds is was 3:1. Mendelගේ cross එකම තමයිනේ. මෙයාගේත් අපි gametes ගමු. මෙයාගේ capital R, simple R. Capital R and simple R. එහෙනම් F2 generation එක කොහොමද එන්නේ? Capital R will be capital R, capital R. That means round. Capital R, simple R will still be round, but it's heterozygous. Simple R, capital R, that means capital R, simple R will still be round, heterozygous. Simple R would be wrinkled, homozygous recessive. In a round to wrinkle seeds ratio 3:1, that is true. Meyage 3 no, meyage 1 no, so that is 3:1. Let's go to the questions. Write the genotype of F1 generation plants. Make a sketch එකක් කොහරි ගහගත්තා නම් ඔයගලන් හම්බෙච්ච ගමන්ම you can quickly just write the answers. F1 generation එකේ genotype එක capital R, simple R. Construct the Punnett square to indicate the genotype of the plants of the F2 generation. So මෙන්න මේකට ඔයා Punnett square එකක් අදින්න ඕනේ. We already know how to draw the Punnett square. Capital R, simple R. Here males lage, then females lage, capital R, simple R. So capital R, capital R, capital R, simple R, simple R, capital R, and simple R, simple R. We have Punnett square again. Same ratios that we got here, we get in the Punnett square as well. Write the genotype of the F2 generation plants. Genotype. Phenotype එක දන්නවා round and wrinkled, right? එහෙනම් sorry, genotype ratio. So genotype එක capital R එක්කෙනෙක් ඉන්නවා. See, capital R. Then capital R, simple R දෙන්නෙක් ඉන්නවා. මෙතනත් capital R, simple R දෙන්නයි. Still round, but the genotype is different. Simple R එක්කෙනෙක් ඉන්න. Simple R එක්කෙනෙක්. That is the answer. Any questions, Lamai? Is everything clear? Were you following? Others? All good? Akib, Dinugi, Oneli, Saritma, Senet, Setumini, is it clear? Okay, I'll tell you how marks are given. Marking scheme. Okay. Superior vena cava, aorta, pulmonary artery, pulmonary veins. May take it okkoma 1 each, 4 marks. 4 no, so 4 marks. 1, 1, 1, 1. C contains deoxygenated blood. Nathang oxygen concentration is low. Hemoglobin 1 mark. D එක ලිවෙන්නේ නැතත් කමක් නැහැ. They're asking the main difference of C when compared to D නේ. now C වල එක ලිව්වා නම් ඇති. Compare C and D කියලා ලිව්වම දෙකම ලියන්න ඕනේ. Here bicuspid valve. You know bicuspid valve is also called the mitral valve. එහෙනම් mitral කියලා ලිව්වත් කමක් නැහැ, but bicuspid is the term that we generally use. Lub sound when bicuspid and tricuspid valves close. එහෙම ලියවනවා 1 mark. Dub sound when semilunar valves close, 1 mark. Make an intervening complete cardiac diastole, atrial and ventricular relaxation. ඒ වගේ තුනෙන් එකක් ලියවනවා 1 mark. Thrombosis, 1 mark. For this 1 mark, for this 1 mark, for this 1 mark, then make it a 1 mark, make it a 1 mark, make it a 1 mark. Punnett square के මෙහෙම gametes ටික හරියට ඇඳලා තියෙනවා නම් 1 mark. Table එකේ ඉතුරු ටික හරියට complete කරලා තියෙනවා නම් another mark, altogether 2 marks for the Punnett square. මෙන්න මේ step එක ලිව්වට 1 mark. නිකම්ම 1:2:1 කියලා ලියන්න බෑ ළමයි. You have to write what is 1, what is 2 and what is 1. So මේක මුලින් ලියලා ඉන්න ඕනේ. මේකට ලකුණු කීයක් හම්බෙලා තියෙන්නේ? Then for this 1 mark. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 16, 17, 18, 19. Okay, Onali. Okay, Ashini. Saripma. Binuti. Akib. Tenumi. Disal. Randira. Dinugi. තුන් දෙනෙක් ඉන්නවා. කවුද? Fatima. Senate. Who else? Batula? Okay, let me know your answers. Okay, we will move on to question number 6. Let me get this question to the whiteboard. Okay, Batula and okay, Senate. You know, at the exam, question number 5 is completely biology. Question number 6 is completely chemistry. Question number 7 is completely physics. Then question and physics. Structured part, okay, question number 1, කොහොමද biosphere වලින් යහන්නේ. Then question number 2 is biology. Question number 3 is chemistry and question number 4 is physics. Okay. Let's discuss the 6th question. Acids, bases and salts are 3 main groups of compounds. Explain what is an acid based on how it behaves in water. What did you write? You have to describe an acid based on how it behaves in water. Water වල යා කොහොමද ඉන්නේ කියලා බලන්න. When you put an acid to water, what will happen? Yes, others? When you put an acid to water, what will it do? Come on, Lama, I only got like a very few answers. What did you all write for this? Well, Q1 2019 full paper karana kira neda? Did I tell you? Did I ask you to do the 2019 full paper? No? Yes, no, ඒක තමයි කිව්වම. So let me tell you 2019 දැන් අපි discuss කරන ගමක් නේ. So rather we will not be able to discuss the entire paper no. Ituru questions whatever and then 2020 full paper, 2021 full paper. Well, I can't know full papers. 2020 full paper, 2021 full paper and 2022 full paper. I Q&A කියන්න එපා. Okay. Explain what an acid is based on how it behaves in water. Acids are compounds that release H plus ions in the aqueous medium. නැත්නම් compounds that release H plus ions කියලා ලිව්වත් ඇති. Okay. Aqueous medium කියලා ලියුවා නම් better, but even if you just write this, it's okay. Roman number 2. Acetic acid, weak acid. What is the difference between a weak acid and a strong acid? You know this very well. What is the difference between a strong acid and a weak acid? Strong acid එකක් නම් මොකද කරන්නේ? Yes. Dissociate completely in water in the aqueous medium. Now dissociate completely into ions in aqueous medium or in water. They can't dissociate completely in water. For an example, if you take HCl, you know HCl is a strong acid. Completely ions will break. So there will no longer be HCl, just H plus and Cl minus, because all HCl will dissociate to H plus and Cl minus. But if you take a weak acid like acetic acid, CH3COOH, මෙයා partially තමයි ionized වෙන්නේ. This also releases H plus ion, so it is an acid. But if you put CH3COOH into water, කොටසක් හැදෙනවා CH3COO minus and H plus වලට. ඊටු කොටස CH3COOH විදියට හැමම ඉන්නවා. Partially තමයි dissociate වෙන්නේ. Strong acid dissociate completely into ions in water, and weak acid dissociate partially into ions in water. Then by what name is the process of forming salt and water by the reaction of an acid and a base known? Acid එකකුයි base එකකුයි react කරලා water and salt හදන reaction එක. Others? Yeah. Neutralization. Roman number 4. Write the balanced chemical equation for the reaction between base sodium hydroxide and hydrochloric acid. Sodium hydroxide, which is a strong base. Hydrochloric acid is a strong acid. You know, මෙතන වෙන double displacement reaction එකක්. We call it a special name. We call it a neutralization reaction. මෙයාගේ Na and මෙයාගේ Cl එකතු වෙලා NaCl හදනවා, which is a salt. Then acid එකේ H plus and base එකේ OH minus එකතු වෙලා හදනවා H2O. H2O. This is the balanced reaction. Write an observation that can be made when the above reaction occurs. Make a venakota, what would be an observation that you can see here? What would be an observation? Observation can anything that you detect from your sensory organs, no? What do you think? Yes. What වෙනමුකුත් පේන්නේ නැහැ ළමයි. NaOH කියන්නේ colorless, HCl is colorless, NaCl is colorless, and H2O is colorless. They are all colorless, so there's really nothing much you can see. Okay. හැබැයි ඔයාලට එක observation එකක් එනවා because neutralization or this acid-base reaction, we call it neutralization. No? This is an exothermic reaction. Exothermic means it releases heat. It releases heat means the vessel will increase the temperature. Okay, you can say increase in temperature. You can say that the vessel is heated. Again, something like a relevant answer you can write. Okay, up to that point, is it clear before we move on to B? Thumbs up. Okay, let's move on to B. Gastric juice contains hydrochloric acid. Antacid tablets given to relieve the discomforts caused by the high acidity. So HCl වැඩිපුර secrete වුණොත් නැත්නම් කැම්බ නැති වෙලට HCl secrete වුණොත් that will create acidity. Inflammation වෙනවා, we call it gastritis. Contains, so antacid tablets. අපි ඒක neutralize කරන්න magnesium hydroxide තියෙන antacid tablets use කරනවා when you have gastritis. What is the salt formed during the reaction between hydrochloric acid and magnesium hydroxide base? So magnesium hydroxide is what is in the antacid tablet. HCl is the acid in the stomach. Again, it is a neutralization reaction. HCl plus antacid tablet එකේ magnesium hydroxide. මෙතන හොඳට valency බලන්න ඕනේ ළමයි when you are writing the reaction. We know acid මෙතන තියෙන magnesium and මෙතන තියෙන chlorine එකතු වෙලා MgCl හදනවා. හදද්දි කොහොමද valency cross වෙන්නේ? Magnesium ගේ valency මොකද ළමයි? What is the valency of magnesium? What is the valency of magnesium? What is the valency of magnesium, lamai? Yes, 2. Magnesium की valency 2. Chlorine की valency 1. එතකොට මේ දෙන cross වෙලා magnesium gets 1 and chlorine gets 2. MgCl2. And acid එකේ H plus and base එකේ OH minus එකතු වෙලා water හදනවා. Now මේක විතරක් මදි, we have to balance this reaction ළමයි. මෙතන Cl2 දෙකක් තියෙනවා, so I will put 2 here. So then there's 2 hydrogens here. මෙතනත් hydrogens දෙකක් තියෙනවා. Then මෙතන තියෙන altogether there are 4 hydrogens on this side, but only 2 hydrogens here. So I will put a 2 here. 2 times 2 is 4. Then මෙතන hydrogens දෙකයි, altogether 4 hydrogens. Here also 2 times 2, altogether 4 hydrogens. Here magnesium එකයි, Cl දෙකයි. Magnesium එකයි, Cl දෙකයි. Oxygen 2. So here also 2 times 1, 2. It's balanced. Okay. Then what is the amount of moles of water formed when 1 mole of magnesium hydroxide completely reacts with hydrochloric acid? Then may balanced. අපෙන් අහන්නේ ළමයි what is the salt form නේ? එහෙනම් මේ reaction එකම අපි ලියන්න ඕනේ නැහැ. මම මේ reaction එක පැත්තකින් තියාගන්නම් අපිට ඉස්සරහට ඕනේ වෙන හින්දා. අපෙන් අහන්නේ salt එක. Salt is magnesium chloride. MgCl2. අහනවා දැන් 1 mole of magnesium hydroxide will produce how many moles of water? For this, we need the balance reaction. What did you all write here? 1 mole of magnesium hydroxide will produce how many moles of water? මෙතන magnesium hydroxide ඉස්සරහ තියෙන 1 නේ. So that means මෙයාගේ 1 mole නම් මෙයාගේ water හැදෙනවා 2 moles. If magnesium hydroxide were 2 moles, water will be 4 moles. If magnesium hydroxide was 3 moles, water will be 6 moles. 1:2 ratio එකෙන් තමයි තියෙන්නේ. අපෙන් ගන්න 1 mole of magnesium hydroxide which will produce 2 moles of water. Is the B part clear to all of you? Give me a thumbs up, please. Okay. Let's move on to C. A bottle contains 500 cubic centimeters of an acetic acid solution. The density of the solution is 1.04 grams per cubic decimeter and the mass of acetic acid contained is 26 grams. Okay. මෙතන ළමයි, how do we produce or how do we form an acetic acid solution? Solution එකක් කියන්නේ homogeneous mixture එකක්. It needs to have components. What are the components in the acetic acid solution? What are the components in the acetic acid solution? What are the components in the acetic acid solution? Yes, acetic acid and water. Methane acetic acid solution එකේ ඉන්නේ acetic acid and water. Yes. So they are saying total volume is 500 cubic centimeters. So මෙතන total volume එක 500 cubic centimeters. මේ question එක sketch කරගත්තොත් හරිම ලේසි. Otherwise it's a bit difficult. Density of the solution. මේ මුළු solution එකේම density එක 1.04 grams per cubic centimeters. That is not the density of acetic acid. That is the density of the entire solution. Acetic acid වල තනි density එක නෙවෙයි, මුළු solution එකේම density එක. මේ තියෙන solution එකේ volume එක. And solution is made of acetic acid and water. Mass of acetic acid here is 26 grams. මෙතන acetic acid වල mass එක 26 grams. Okay. I kind of have a full picture now. Calculate the mass of acetic acid solution. Now, acetic acid වල mass එක තියෙනවා නේ? We have the mass of acetic acid. Now they are asking us the mass of the acetic acid solution. Can you see? අපිට පුළුවන් නේද density equals mass over volume කියන equation එක use කරන්න. Because we have the density of the acetic acid solution. මේ ඔක්කොම values ටික අපිට solution එකට තියෙනවා, not just acetic acid. I hope you understand when I say acetic acid and acetic acid solution. They are two different things, right? Acetic acid water වල mix කරාම තමයි acetic acid solution එක එන්නේ. Then අපිට solution එකේ density එක තියෙනවා which is 1.04 grams per cubic centimeters. අපෙන් අහන්නේ solution එකේ mass එක. Solution එකේ mass එක. Okay. අපිට solution එකේ volume එකක් තියෙනවා 500 cubic centimeters. එහෙනම් ළමයි අපිට මෙතනින් ලේසියෙන්ම mass එක calculate කරන්න පුළුවන්. So mass would be 1.04 multiplied by 500. That would be 520 grams. May zeros දෙක cancel වෙලා decimal point එක යනවා. So 5 times 4 is 20. 5 times 1 is 5. So you get 520 grams. Is this part clear? We'll go step by step. Is this part clear? Others? Okay. ඔන්න එහෙනම් අපිට මුළු එකේ mass එක, not the acetic acid mass, I have the total mass. Acetic acid plus water, total mass එක ඔන්න 520 grams. Okay, let's move on to the next question. Calculate the percentage of acetic acid by mass in the above solution. Acetic acid වල mass එක කොච්චරක්ද? Acetic acid 26 grams තියෙනවා. Percentage එක නහන්නේනේ. Acetic acid වල mass එක 26 grams. Total mass එක 520 grams. Percentage in I multiplied by 100%. So grams cancel වෙනවා. 26 times 120, 26 times 2, 52, මෙතන 0 එකක් යනවා. 20 times 120, 20 times 5 is 100. So you get the answer as 5%. And acetic acid වල mass එක as a percentage is 5%. Vinegar is an aqueous solution which contains 5% acetic acid by mass. දැන් අපි කතා කරපු එක්කෙනා වගේ. By boiling, the boiling point of acetic acid is 118 degrees Celsius. Name a technique that can be used to obtain a solution that contains about 10% acetic acid by mass using a sample of vinegar. Okay, so අපි මොකද්ද කරන්න ඕනේ? 5% mass එක තියෙනවා acetic acid, that is vinegar. මට මෙයාව convert කරන්න ඕනේ 10%. ඒ කියන්නේ ළමයි acetic acid concentration එක වැඩි වෙලා නේද? The amount of acetic acid compared to the total mass has increased. ඒක තේරෙනවද ඔයගලන්ට? අපි percentage එක හෙව්වේ මෙහෙමනේ. හොඳට අහගන්න. We found the percentage. Percentage equals mass of acetic acid we divided by the total mass and then we multiplied by 100%. දැන් ඔයාට vinegar sample එකක් තමයි දෙන්නේ. vinegar වල 5% acetic acid තියෙනවා. Right? ඒ sample එක විතරයි use කරන්න ඕනේ. You can't use anything else, just that sample. You have to convert it to a 10% acetic acid solution. ඒ කියන්නේ percentage එකම වැඩි කරන්න ඕනේ. percentage එක වැඩි කරන්න නම් මම මොකද කරන්නේ? How do I increase the percentage? What do you think? So percentage එක වැඩි කරන්න පුළුවන් මට. I have two methods. එකක් තමයි I can increase the mass of acetic acid solution. If I increase the mass of acetic acid solution, the percentage of acetic acid will increase. ඒක තේරුණාද ළමයි? This is a very important question. මේක ගොඩාක් අයට හරි ආන්සර් එක ඒ අවුරුද්දේ ඇවිල්ලා තිබුණෙත් නැහැ. ගොඩාක් අයට මේක ඊට පස්සේ කරද්දි තේරෙන්නේ නැහැ. ඉතින් හොඳට අහගන්න. Percentage එක වැඩි කරන්න මට එකක් පුළුවන් නේද? I can increase the mass of acetic acid in this solution. Is that clear? Thumbs up. Okay. But there is a problem. ඔයාට already vinegar එකක් දුන්නානේ. Already vinegar එකේ තියෙන acetic acid ඔයාට වෙනස් කරන්න බෑ. So ඔයාට mass of acetic acid වැඩි කරන්න නම් you will have to add more acetic acid into the solution. But you can't do that. තියෙන vinegar එකෙන්ම තමයි කරන්න ඕනේ. එහෙනම් මේ method එක හරියන්නේ නැහැ. තව එක option එකක් තියෙනවා. percentage එක වැඩි කරන්න නම් total mass එක අඩු කරන්න ඕනේ. If you decrease the total mass, lamai percentage will increase neither. Is that clear? Thumbs up. Others? In order to increase the percentage, another thing you can do is decrease the total mass. Anita, Randira, Dinugil, Saritma, Setumini, Senet. Total mass එක අඩු කරන්න නම් now total mass means mass of acetic acid plus mass of water. මට total mass එක අඩු කරගන්න පුළුවන් විදියක් තියෙනවා mass of water අඩු කරොත්. If I want to reduce the mass of water, මට කරන්න තියෙන එකම දේ water අයින් කරගන්න එක. මේ acetic acid solution එක ළමයි. This acetic acid solution which contains acetic acid and water. මේ solution එක මම heat කරනවා 100 degrees Celsius වල. Now they have given that the boiling point of acetic acid is 118. So කොට ප්‍රශ්නයක් නැහැ නේ. මම 100 degrees Celsius වලට රත් කරද්දී water විතරයි boil වෙන්නේ. Acetic acid will not boil. And when I heat it to 100 degrees Celsius, water will boil and water will form vapor and leave the solution. හැබැයි 118 වෙනකන් රත් කරන්න එපා. එතකොට acetic acid උත් යනවා. I don't want to reduce the mass of acetic acid. I want to reduce the mass of water. So I'm going to heat it to 100 degrees Celsius. Then this will boil and water will vaporize. එතකොට මොකද වෙන්නේ? Mass of water අඩු වෙනවා. Acetic acid, nothing happens. Mass of water අඩු වෙනවා. Mass of water අඩු වෙනවා කියන්නේ total mass එකත් අඩු වෙනවා. Total mass එක අඩු වෙනවා කියන්නේ percentage එක වැඩි වෙනවා. So this is what you can do for this particular situation. So Roman number 3 නේද? Roman number 3 එකේ answer එක හරිනම් heating to 100 degrees Celsius, but even if you write question එකේ කොහොමද දෙන්නේ name a technique එකක් නේ අපිට කියන්නේ අපිට use කරන්න පුළුවන් technique එකක් අහන්නේ. So technique එක නම් the best answer would be vaporization. Vaporization තමයි best example එක. මොකද එතකොට water ටික යනවා. එහෙම නැත්නම් ඕගලන් කැමති නම් distillation කරන්නත් පුළුවන්. හැබැයි simple distillation වලදී එතනත් ඉතින් අපි ටිකක් අයින් කරගන්න පුළුවන් නේ. So එතනදී 100 වල රත් කළා water ටික අයින් කරනවා. Then ඉතුරු වෙන්නේ acetic acid. So these are the answers. Up to that point, is it clear, lamai? Thumbs up. Akib, okay. Dinugi, Fatima, Saritma, Senet, Setumini. Okay. Let's quickly discuss D and then finish the class. D. මම මෙතන D discuss කරන්නම්. A set of apparatus arranged by a student to prepare a sample of hydrogen gas using dilute hydrochloric acid and zinc. Okay. මොනවාද හදන්නේ sample of hydrogen gas. Write 2 errors that can be seen in the above setup. මොනවාද errors? What are the errors? Yes. One is correct. Conical fissile funnel එක මීට වඩා immerse වෙන්න ඕනේ. otherwise මෙතන හැදෙන hydrogen gas ටික මේක දිගේ විතරක් නෙවෙයි thistle funnel එකිනුත් එළියට යනවා. එතකොට අපරාධය. Therefore, yes, one of the mistakes is thistle funnel is not immersed. One of the mistakes is thistle funnel is not immersed in the solution. They're asking two errors. What is the other error? Yes. ළමයි hydrogen gas හැමලෙම උඩටනේ යන්නේ. මෙහෙම hydrogen gas collect කරොත් will the gas be collected here? Will the gas be collected here? No. Why? All the hydrogen gas will go up because hydrogen has a lesser density. This method would be good to collect carbon dioxide because carbon dioxide වල density වැඩි හින්දා එයා හැමලෙම පල්ලෙහාට යනවා. එතකොට ඉතුරු air particles ටික උඩට යනවා. We call this upward displacement of air. But here hydrogen ගත්තොත් hydrogen හැමලෙම උඩට යන්නේ. එහෙනම් අපි මෙහෙම ඇල්ලුවා නම් හරි. එතකොට hydrogen ටික උඩට යනවා. ඉතුරු air ටික පල්ලෙහාට යනවා. That would be downward displacement of air. But here this is wrong. So the other error would be keeping the gas jar upright. හෙලින් අතර තියෙන එක තමයි ඊළඟ error එක. Okay. Roman number 2. Suggest two measures that can be adopted to increase the rate of reaction or taking place in the conical flask. Conical flask के rate of reactions කොහොමද වැඩි කරන්නේ? Rate of reaction වැඩි කරන්න පුළුවන් number 1 surface area වැඩි කරලා. මෙතන zinc granules වෙනුවට ඔයා zinc powder වගේ use කරොත් we would be able to increase the surface area. Using zinc powder instead of zinc granules. එහෙම නැත්තම් මොනවද අපිට කරන්න පුළුවන්? Concentration දාන්න එපා ළමයි. මොකද මෙයා dilute hydrochloric මේ acid එක ඒ විදිහට use කරලා තියෙන්නේ. So there needs to be a certain concentration. Ekina concentration එක better to use other answers. Concentration එකත් it's okay. But අනිත් answers වලට main focus එකක් දෙන්න. So increasing the concentration we'll just write because that is one of the things that we have learned. Increasing the concentration of the acid of hydrochloric acid. I applied temperature. So you can increase the temperature and increase the rate of reaction. So yes, increasing the temperature or heating. Heating the conical flask. All the other method that we have used is last method, which is using a catalyst. So you can use a catalyst. So මේ වගේ මේ තුනෙන් මේ හතරෙන් මොකක්ද දෙකක් clear තියෙනවා you get marks. එම නැත්තම් තව එකක් ඔයාලට පුළුවන් ළමයි this conical flask you can make it smaller. පොඩි conical flask එකක් නම් එතකොට ඔක්කොම එකට එකතු වෙලා වැඩියෙන් ඉන්නේ. Right? That means when there's a smaller conical flask, the space is restricted. So all the zinc granules and dilute hydrochloric acid has like more probability for interactions. The collisions වැඩි. Conical flask එක පොඩි වෙනවා කියන්නේ collisions වැඩි. ඒකත් ඕන ලියන්න පුළුවන්. Okay. State a test and the relevant observation to confirm that the gas produces hydrogen. Make a test එක මොකක්ද? Introducing a lighted Eagle or splinter. What is the observation here, ළමයි? What is the observation here? Yeah, the gas will burn with a pop sound. The gas will burn with a pop sound. Or just burns with a pop sound. Up to this point, is everything clear, ළමයි? Before I go to marks, is everything clear? Okay. Marks වලට යමු. Here for this 1 mark, 1, 2 marks altogether. For this 1 mark, reactants stick at a 1 mark, products stick at a 1 mark. For this 1 mark, then make it a 1 mark, make it a 1 mark. No, equation එක අපෙන් අහලා නැහැ, moles නේ අහලා තියෙන්නේ, so make it a 1 mark. Then here, calculation එකට 1 mark, getting the answer as 520 grams, 1 mark. Here again for the calculation 1 mark, getting 5%, 1 mark. Here for writing the technique, either vaporization or distillation, simple distillation, 1 mark. Here, for 1 for each, 2 marks. Here make it a 1 for each, 2 marks. දෙකයි නහන්නේ නේ. Here for writing the test, 1 mark. For writing the observation, 1 mark. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Okay. How many marks did you get out of 20? Alamai? Okay. Okay. Others? Okay. So we will end the class for today. I will let you all know the reschedule classes for the prashna, since you have exams and school. But I will discuss and let you know. Then until we meet the next day, have a great week, study well. And if you have any doubts, any help you need, make sure that you let me know. Okay. So see you the next day.